The culture techniques, the clinical, we were studying just earlier, and we covered the microscopy, and then we are coming towards the culture techniques. Culture techniques difference means that we are growing bacteria in a lab setting, but not just bacteria, microorganisms, of course, all everything. So, for example, growing them in a liquid media. Media is actually the material that has nutrients and is used to grow the microorganism, and it actually has everything packed in it which the microorganisms would need. So in culturing techniques, there are a lot of techniques like plate method, plate spreading method, enrichment culture method, streak plate method, and things like that. Or maybe culturing on the solid media and culturing on liquid media, or culturing in suspension media, or serial dilution methods. If you wanna study them more, you can comment below.